Hello and welcome to the September Getting Started with iPlant webinar. My name is Jason Williams and I'm coming to you from Cold Spring Harbor Laboratory, New York. I'd like to say welcome. This webinar goes out to the several hundred people who sign up each month for a new iPlant account. As you see from the survey and from many other surveys, a lot of the times it's by word of mouth that people find out about iPlant and want to start using it, want to start analyzing their data. And so we put together this 1-hour presentation to help you get started, know what some of the capabilities and features of the platform are, and also answer questions. So we'll be together for about the next hour or so. Fortunately, the entire video will be recorded and posted to YouTube not too long after, so you'll get that notification, and then if there's anything you missed during the presentation, or if there's anything you wish to share with others, you can show them the recording of the video. Before we actually get started with the main presentation, let me tell you a few features of the Adobe Connect webinar that we're using in order to transmit the program. So first of all, at the bottom of the screen, there is a place for you to ask questions. So at any time off screen here, I can actually monitor who's in the room and who's asking questions. So if you do have a question, please at the bottom, go ahead and chat that at any time, and I'll respond to the question and try to answer your query. Also, off to the side of the screen, on the side there are a couple of features, including the ability to download the entire presentation. So there's a PowerPoint that's there, also some getting started guides, cheat sheets, etc. Also some important links, so feel free to use any of those throughout the webinar. At the top of the screen, there's also some buttons on the video screen itself. One of them, those 4 arrows that you see in a little box, that allows you to make the video full screen. When I go to show you some of the things on my own computer, it probably will be important for you to make it full screen so you can really see some of the details. You'll have a much better way to view those. Also, if you move your mouse over the video itself, you can adjust the quality of the video. So if perhaps you are on a low bandwidth connection, you might want to change the quality of the video to a little bit lower in case anything looks choppy. Other than that, I think we're ready to get started, and like I said, I can see those questions off to the side there, so if any time, please interrupt me and let me know what kind of questions you might have. So we have a couple of different aspects to this presentation. At the beginning, I'm going to give you a brief overview of exactly what is the iPlant Collaborative, what do we as a project produce and have available to you, and then I'm going to give you a few hands-on demos to some of the more popular platforms that you're likely to use in your first experiences with iPlant. So let's get started with the presentation. It's really important for me to convey a little bit of the background of what the iPlant Collaborative is as a project. Okay, so let's get started. The first thing about iPlant is that we as a project have our own vision. This is really important because it guides the decisions that we make in terms of what do we produce for our users. I've taken this little take on it. I've got 2 images here. One on the left-hand side is a cartoon from The New Yorker. As I said, I'm based in New York. And it's actually a little cartoon of a famous Italian explorer, Verrazano, who's looking over the cliffs of the Hudson River saying, someday this will all be infrastructure. And as you know, later on, of course, in New York City, certainly a place for bridges, roads, tunnels, infrastructure. You can build a city. But in this case, I have him sort of staring at the double helix image that we all know from Rosalind Franklin that gave rise to our understanding of DNA as it is. And the question is, well, really, someday that double helix will all be infrastructure. When we're talking about working in the lab today, we're not talking about, most of us, necessarily doing hands-on biology, but really it's moved beyond just working with test tubes to now working with data sets, working with computers, computation. And so how do you prepare for something like that? How do you actually make that leap from being a hands-on biologist to being a bioinformatician? And even if you're already into using bioinformatics, as a lot of you are in many aspects of your research, how do you prepare for things that you can't anticipate? So that's the vision of the iPlant project itself, and it guides our decision-making process. We're always trying to think of what our users will need down the road, and we're always collecting input from our users so that we have what you need when you need it. So what is that vision? Let's continue with the story. Well, as we know in the case of Verrazano, it was building a bridge, and for us, it's building something called cyber infrastructure. So our vision, put into sort of words, is to enable life sciences educators and researchers to use cyber infrastructure to understand and even ultimately predict the complexity of biological systems. And that means looking at biology not just as biology unto one of itself, but also biology and its dynamism, considering environmental conditions, so genotypes, phenotypes, environments, all of those things are brought together because when we're asking large questions in biology, we have a lot to consider. We have all those domains that we need to be informed about. But let me define this word a little bit more explicitly, cyber infrastructure. Cyber infrastructure is nothing you don't already use to some extent. It's data storage, software, high performance computing, and even people. But what's key about cyber infrastructure is that it's organized into systems that allow you to actually solve biological challenges. So it's putting together the pieces in a way that really lets you get your science done. Our cyber infrastructure project is tailor-made for the types of questions that biologists will want to analyze, will want to answer. So putting those pieces in the right order, in the right system, really makes a difference, as I hope I'll show to you. When you think about the products that we produce as a project, this is a sort of a layer cake diagram of some of the projects. In fact, we'll be talking about quite a few of them, from cloud computing, graphical interfaces, educational interfaces. All of these products that you as the end user are going to use on a day-to-day basis, this is what we'll be considering in the demo. You'll see these to some extent. But underneath, there's a lot of this cyber infrastructure aspects that you won't necessarily see, but are nonetheless quite important. Things like being able to store the data, interact with it through APIs, security, authorization, being able to log into your data sets and know that they are secured, being able to search those data sets, and then all the way at the lowest layer, access to the actual systems, so high performance computing, database, cloud systems, all those different things make up what's underneath the hood, so to speak, of the iPlant products. Put simply, or put another way, cyber infrastructure is really access to all of these things. So platforms, tools, data sets, storage and compute, and then training and support. And so, of course, today is an example of that sort of people aspect of cyber infrastructure, getting the support you need. So the next thing is that a question that we have to ask is, what are the kinds of problems that iPlant can solve? Well, as you might understand or infer from the name, we certainly work with crops, model plant systems, non-model plants, but we have users in all domains of life science. So animals, livestock, agronomic microbes, insects, really iPlant is built for data. So any type of life science problem, project that you're working with, the data aspects of it are certainly can find various solutions to in iPlant. How is iPlant built? This is also important for you to know. Really, at the beginning of it, we knew that we couldn't just come up with all the solutions ourselves, but really it was going to be a community project. So when we spent almost the first year and a half, almost 2 years of intensive requirements gathering, what we learned from the community is very much what I think you'd expect. Genomic data has definitely been the big driver of data-intensive biology. So when you come to iPlant, you will definitely find tools for doing genome assemblies, RNA-seq, variant calling, ChIP-seq, a whole entire platform set up for working with genomic data. But one thing that very much distinguishes iPlant is that we've never been solely about working with genome data sets. If you look, really biological problems involve genome data, environmental data, phenotype data, to name 3 large categories. And it's by combining those data sets that you can really make larger inquiries, larger projects, larger questions, larger investigations that often deal on higher level organization, evolutionary, ecological models, association studies, looking at pathways. So really these are the priorities that iPlant is concerned with, and we do have project systems, tools, expertise in a variety of these domains. So it's not just about genomic data. Now, if you want to see where we're at, because we're certainly not done in the things that we'd like to deliver, you can always check our website, but this diagram gives you a little bit of an idea. Certainly, I think for genome data sets, we have the foundation in place for just about everything that we've been requested to do. And environmental and phenotypic products, we have many platforms in place, many things that are undergoing evolving deployments of new technologies, new pathways in terms of workflows. So really stay tuned to our website, because that's the best place to get updated information as to what's available and also what's coming. Who does iPlant collaborate with? This is important also because iPlant is not just about believing that we're the only solution to every problem that you're going to have. Certainly there are many different projects that are out there that cover aspects, sometimes overlapping, sometimes not, with what iPlant offers. And so our goal as a project is to make it easy for you as the user to move your data and analysis wherever it needs to be. So if there's some solutions that are available only in K-base, for example, we want you to have interoperability between those tools and data sets. But if you need to move the data around, we want you to be able to do that. We never want to tie people in to somehow only using iPlant. And on top of that, iPlant is completely open source. So really, anything that you use with iPlant, you can be comfortable that you're not somehow locked into something that you must exclusively use it. But we really want you to figure out iPlant as to what makes sense for you and use those parts that are going to work best for you. So the collaborations is certainly our way of doing that, by working with everybody that's out there that has something to offer. Another important aspect, who makes up iPlant? Well, we ourselves are a collaboration. So between the University of Arizona, University of Texas Austin at TAC, Cold Spring Harbor Laboratory, UNC Wilmington, we're a collaborative and entirely virtual organization. We're funded entirely by NSF. This is important because it means that over the past several years, since our initial funding in 2008, we've become a $100 million investment. So really, these are resources that are paid for, they're on the table, and they're open to you. And that also goes for users not only within the US, but your collaborators. So you may have people that are not in the US. We really invite everybody to take advantage of these resources to advance science because they are there for you. And really, what we hope to show you today is some of the abilities and functionalities that you can take advantage of right this second. In our renewal process, it's also worth mentioning, we also have a scientific advisory board, and it's with their advice that we listen to the community, and we of course take your direct input in order to shape what we deliver. And we also have really a great focus on genotype to phenotype science. So that's our interest. And again, as you saw earlier, we've anticipated that because it really means not just genomic data sets, but the whole landscape of the different biological data types that our users are interested in. Okay. So I've given you really quite a bit of background, but let me just quickly take you into what some of the advantages are of working within a unified platform. Number one, at the end of the day, you want to get your science done. And so what we think that means is the ability to access and manage your data sets, the software you're going to need in order to do the analysis, the computing resources, as well as skills and help. Another important thing is reproducibility. This is extremely important for people who are who new perhaps to their first bioinformatic project, big computational project, being reproducible. This can be difficult sometimes, and we know, reading some of the literature, that this is a problem in various areas of computational science. So working within the iPlant platform, you're given really the ability to manage the metadata, to share data and workflows, and again, it's all within the context of open source tools. That means that you really have a lot going for you when you want to reproduce that analysis. When you publish it, you know that other people will be able to perform the same analysis. A final major point that I'd like to pull out is productivity. And certainly by working within an environment that gives you high performance, scalable computing, you can get things done a lot faster. We can enhance your ability to automate and collaborate. And also that means more of your funding can be spent on science, not on software and hardware. So those are really some of the major advantages. What I'd like to go into now, after I've given you a really quick introduction, but I hope one that gave you some perspective onto the background of what iPlant is as a project, what I'd like to talk about now is what do you actually get with your account. So once you sign up, you may have signed up for a certain reason, someone told you that you need to do this or that, but I do want to make sure we give you at least some type of overview as to what comes with your account, what do you have access to. Okay, so remember my sort of analogy taken to the next step here. We think of ourselves almost as the sort of the building bricks or the Lego blocks of cyber infrastructure. We have built 85, 95% of what you want, but in almost all situations, you're going to want something that's a little bit custom because your data set is custom, your question is unique. So we've put together most of the pieces, you assemble them how you need it, and if the solution is not available directly, out of the box, we can help you build that. In many cases, you can build it yourself. let's talk about those pieces. So first is the iPlant data store. So every user, as soon as you sign up, you're granted a 100 gigabyte allocation. That's not to say that you might not need more than that. And of course, if you do, you may need even terabytes for some larger projects. That data storage is available to you. We do request that you just complete a little form to let us know the justification for it. The data are automatically backed up, which is great for a data management plan. You can know that the data is secure and it is backed up continuously. And we've made it relatively easy, I think, to upload, download, and share those data. And I'm going to be showing that for the first part of my hands-on demo. The discovery environment is really important for the majority of bench biologists who may not come with a command line computing background. It's an environment where you can use hundreds of bioinformatics applications in an easy-to-use web interface. You can run just about any bioinformatics application that would normally be run at the command line. It's already integrated with our data sets and compute, and it's user extensible, which means you can bring your own applications and run them on the discovery environment. Atmosphere is our cloud computing solution. If you've never used cloud computing for science, we'll explain that during the demo and show you how easy it is to get started. But it's access to hundreds of virtual machine images. You can customize your software setup, and it's already integrated, as I said, with just about everything else in iPlant with our computing and data resources. The science APIs are very interesting for those of you who are developers or programmers. We won't be covering this in this demo, but you should know that they do exist. You can essentially use them as a science as a service platform to define your compute and storage resources and to mix and match those, and also to build your own app store, scientific codes, and workflows. And there's documentation available on the website. Another thing we won't be covering today, but which is also a fantastic tool, is DNA Subway, which is our educational workflow for genomics, DNA barcoding, RNA-seq, and it's really a way to bring bioinformatics into the class form. We have a number of workflows that are, I call them classroom-friendly bioinformatics workflows, plenty of sample data sets, and also hands-on laboratories and documentation there if you want to use bioinformatics in the classroom. BISC is our image analysis management platform, where you can work with image data, store those data, search. You can have custom APIs and analysis routines, so it's great for looking at phenotypic data of many scales, hundreds of different data formats, image formats supported there. And that's a quick overview of those products. So again, we're not going to get to cover every single one of them in the course of this hour. It would be way too much. But we will show you the iPlant Learning Center and other resources online so that you can at any time go ahead and investigate any of those that we don't happen to cover today. Okay, so my first demo is going to be on data. Let me remind you again at the bottom of the screen, any questions, please go ahead and chat those. This is the time during this 1 hour webinar to have somebody live trying to answer any questions, but of course I'll show you later where you can go online and post any questions that you might have after, or of course watch the recording of the video. So let's talk about our first demo, which is getting data into iPlant. That's the first step. Okay, so as I mentioned, we have the iPlant data store. You can store any type of file related to your research. You can move the files seamlessly between the different iPlant platforms. You can automate file transfers. You can share those files with lab members, with collaborators, with communities, the public sort of sites that we have. So what we're going to focus on are some of the ways to access. Now, there are multiple ways you can access the data because we have multiple use cases for what our users would like to do. So I'm going to focus on one point-and-click method, which is CyberDuck. It's a really unique and interesting tool. It works very much like Dropbox. It's going to be super user-friendly. So we're going to show you a demo on that. And we'll go back to the discovery environment later because that's really a great platform for sharing the data. I'll also briefly show you the command line because there are some users who are familiar with that and would like to know right away how to be able to get their data moving through that system, through that interface. In the discovery environment, as I mentioned, you can work with some smaller files. You can import from URLs. The advantage is it's really easy. It covers some of the day-to-day uses of working with the data sets, although there's some speed and size limitations. CyberDuck is really going to be the solution for most folks because it's drag and drop. It's more like I said, compare it to Dropbox. The iDrop desktop is available. It's something that we had supported in the past. It's not something that we're going to cover today, but it's also another graphical interface, and you can use that as well, including working with permissions. And iCommands, I will show you, this is for the command line, and it really gives you full flexibility. Okay, some of these require Java, the one in the discovery environment, but that's not really going to be a concern for us today, so we'll go ahead and get started with this in the demo. The first thing we need to do is actually see where everything is, so I want to encourage you to go ahead and even bookmark the iPlant Collaborative homepage. We'll keep coming to this a couple times, so from here we can access just about everything that we need. If you scroll down to the right-hand side, there is the Learning Center, and I'd like you to go to that first link that says Get Started. Again, you don't have to follow along with me, but you certainly can watch the video later. In the Getting Started section, I'm just going to skip ahead to this section that says Import Data. It's the Import Data section where we can actually go to download CyberDuck, and it's available there for Windows and Mac. You're actually going to have to download two things. One, you're going to have to download the CyberDuck program, which is here, and then you're also going to have to download the CyberDuck profile. So this is like a little file that allows CyberDuck to sort of log in. Now I'm going to click on the documentation just so everybody sees what it looks like, okay? Because you're really going to want to follow the documentation to install it the first time. But it really takes less than 2 minutes, I would say, to get it running. Everything is step by step with screenshots. Now I have CyberDuck running. And I'm sorry I can't make it look much bigger, but this is the time of the demo. Remember I said on the top of your screen you have those arrows. You can make the video full size. You might want to do that right now. Okay, so I've opened a connection. In fact, if I go ahead and open another one, you could see what it looks like. Let me see if I can start another instance here of CyberDuck. I'm not sure if it will. I'll show you that second. But what I'm looking at right now, these folders, are actually folders and files that are in my iPlant data store. So essentially, once I connect to the iPlant data store, I see them. I can pick a file that I think is going to be pretty small. In fact, this one is a few kilobytes. And all I need to do if I want to download that is drag it over to my desktop. Okay, and then I'm going to see here a little transfer dialog that pops up. In a few moments, that will start, and the download will continue going. So it takes a few moments for it to log on, but essentially that will go. And you can move large data sets. So we've done this testing with, I would say, several hundred gigabytes of data, and that's really no problem to transfer via CyberDuck. The same way, if I had a folder that I wanted to move, I'm not going to move this whole entire folder, but let's see, here's something small. I'm going to pick a folder, and I want to upload that into my iPlant data store. It would be the same thing. I drag it over into my iPlant data store, and then I would see that happening. Okay, the first one has completed, so that file that I downloaded, FASTA file, some proteins it looks like, has been downloaded, and then the other one will upload. So it takes a few moments to start the connection, but once it goes, it's quite fast. Let me show you again from the beginning. If I was not connected to iPlant, what it would look like. I'm just going to disconnect, see if it lets me do that with a transfer in progress. I'm going to go ahead and quit. So when you download CyberDuck for the first time and install it, you're just going to download that profile, and then the very first time you run it, where it says data.iPlant collaborative, you're just going to go ahead and click on that. It takes about 2 minutes in my experience for it to sort of log on to the system, but once it does, you're logged on, and really moving large sequence files, for example, is then just a matter of dragging and dropping, so pretty simple. Okay, let me show you moving those files back and forth at the command line via iDrop, or rather, I'm sorry, iCommands. So back on the iPlant website, let's go back to where we were in the Learning Center. There's also the download here for iCommands. So again, if you're working Mac, Linux, you can download those right away and view the documentation. I've already got iCommands installed on my computer, so I'm going to go ahead and start that. And let me open up a shell that's easier for you to see. Ooh, I wanted to make one that's, there we go, perfect. And this one I can make a little bit bigger. And essentially, once I have iCommands installed, all right, it lets me know, here I am connected to data.iPlant collaborative. This is my, my iPlant username. If I want to view the contents of my data store, it's like Linux command, so an ls. Instead of ls, it's ILS, hence iCommands. Will let me view the contents of my iPlant data store. If I want to download a file, I can use an iget command. I'm going to say I'm going to download a folder, so I'm going to make it recursive. I want to see the progress, and then I want to do VCFs, and I'm going to do that to my desktop. Okay, and then on my desktop you can see that folder has appeared, and then here it's going to show me the progress for that file transfer, and that will take a few seconds. So really, I've shown you now two methods for transferring data, one slightly advanced, one quite easy, user-friendly for most of us. The CyberDuck, go ahead and download that, and you really will have drag and drop access to your data store right away. You can start moving files into iPlant. If you prefer the command line, go ahead and download iCommands, get those installed, follow the documentation, and then you can quickly automate, build scripts against, do whatever you'd like to do with the transfer of data in that way. Okay, so now let me show you how to work with sharing those data. So I'm going to go back to the iPlant homepage. Okay, at iplantcollaborative.org. Okay, so back at iplantcollaborative.org, what we're going to use to actually manage the sharing of the data is a discovery environment. So all you need to do when you move your mouse over there, you can see a button that says Launch. You can go ahead and click on Launch. I am already logged in. You would probably have to log in with your iPlant username and password. Now this is the discovery environment. We're going to be talking about its features in just a moment. But right now, let me just focus on data, which is where all the data that I have stored within iPlant is located. If I click on my username, I see the same data sets that I saw just a moment ago using CyberDuck or also using iCommands. I can scroll through this, I can drag and drop folders, I can rename things. So it works very much like a desktop application, many of the things that you'd be expecting. But the key feature that I'd like to share right now is how do I share a data set? So suppose that I select a folder and I want to share this folder with someone else. I can go up to Share and then share an entire folder with another collaborator. So this person must have an iPlant account. I'm going to show you a different way later. But if I go ahead and say Share with collaborators, I have a folder. This one I'm pretty sure is shared with a couple of people, and it's got several gigabytes, I think maybe a little bit less than 100 gigabytes of data in it. But I can instantly give anyone access to it. So if I want to do that, I can search for a user by typing in their name. As I start typing in their name, I see the user and their affiliation, and then I can decide what level permission. These are described in documentation, but essentially with read permission, that person can only see the data set. They can also analyze it, but they couldn't move it or copy it or do other things. If I give them own permission, they can do everything, including delete it, so I have to be very careful. But essentially, if I were to click Done, that person would get a notification that the data set has been shared with them, and they could go right to it and see it and start using it. If I shared something with somebody and I no longer want to share it with them, I could come back to it and click the X. And now I've revoked permission. They'd no longer be able to access that data. So really, sharing is as simple as selecting a folder or selecting a file, going up to Share, and then say Share with collaborators. Now in this case, they must have an iPlant account. There is a way to share without having an iPlant account. This way only works with individual files, so you could either share a small file or you would have to zip up something into a file. I wouldn't recommend that you share things that are extremely big this way, so several hundred gigabytes. Even above 10 gigabytes would not be the best way. But in any case, for a simple file, I don't want them, they don't need to have an iPlant account, and go up to Share and say Create Public Link. And I can click on that file and say go ahead and create. And now it would create a URL that I could give to anybody and they would be able to share it. I'm going to go ahead and copy that link, Control C. And now when they go and open up a web browser, they could click on that and then they would be able to download that file. Okay, excellent. All right, if I no longer want that link to be active, I would need to come here and delete that link. Be careful, as it says right there. Once I create a link to a file, anybody who has that link could share it. So if I needed that to be secure, this might not be the best way. But for just a quick share, that's fine. Here I can go ahead and delete that data link, and now that's no longer shared with them. Okay? Data sets that other people have shared with me are over here on the left-hand side under Shared with Me. When you go there, if you log in the Discovery environment, you'll see a variety of folders because there are some data sets that are shared with everybody in iPlant, but you'll also see things that people have shared with you. Okay? In community data, which we'll use in a moment, there's some shared data sets. But in your own folder, you definitely have the ability to create folders and files that you can share with anybody. You might want to create, for example, a shared folder with your entire laboratory where you have one folder, everybody deposits into that. You configure permissions. So it's really an excellent way to share data sets. Once you've gone through that initial upload into iPlant, it's there. And so for them to see it and use it and start analyzing it, it's instant. Okay, so to come back to the screen here, the only other thing I could show you is at the top, you can also search for data sets. If I search, just by simple plain text name, I could see, I'm searching for genus, which I know at least some files might be named that. I could do a more advanced search because every file in here, let's see, I'm not sure about, this particular one, but if I click on it and go up to edit and say share metadata, let's look at the metadata for this file. This one doesn't have any metadata attached to it, but I could add metadata like species, and I could say, or rather, let me say genus, and I could say Dioscorea, and say OK. Okay, now I've added that metadata. Now if I go up to a smart search, I could look for metadata, has the attribute genus, has the value Dioscorea, and I could search for that. And now anything that matches that, now this will take a few seconds, so I wasn't expecting it to find it that second because it's still updating. But if I look to, I think I have some other things that will come up in search if I remember any of them, which I'm not at the case, but you can actually search by that. Whether the genus or the value or species, you can essentially create your own sort of smart searches and filters with this. Although I wasn't successful today because live demo, but no problem. All right, so that's all that I wanted to show you about data. Now let's go to the second part, which is how do we actually analyze that data once we get in there. So far we can upload and download data. We can share that data with any colleague or even people outside who don't have an account. We can even add some metadata to that, and maybe I'll show you a little bit more on some things that I know have. But you can do a lot with data now, even just looking at that quick demo. How do we analyze the data? So I have a couple more slides to talk about the DE, and then we'll do a very simple analysis. Okay, so let's go back to the presentation. And I've got a whole bunch of windows open now, but here it is behind. All right. All right, so getting started with the discovery environment. The discovery environment, as I mentioned, is a platform, so you can run almost any bioinformatics application. It's integrated with our data and high-performance computing, and it's also user-extensible, which means you can add your own applications. I won't show you today adding your own applications, but it will at least get to analyzing some data sets. From the data window, as we already saw, you can upload and download files, you can share URLs, you can share files and folders. We're going to be using the Apps menu, which contains a listing of the applications within the discovery environment. You can also build workflows, which I won't show you today, and you can actually add applications. Again, those two are covered on YouTube and also in documentation. And then you can also monitor the status of your job. So we're going to go into those other two aspects there, the discovery environment. You can always see a current list of what applications are installed at this link through the iPlant wiki. And now we're going to do a very quick demo. This demo is just going to be, we're going to take some unaligned DNA sequence and we're going to align it using muscle, multiple sequence aligner. So a very simple task, but once you understand how this works, you'll basically understand how all the analyses within the discovery environment work. First, let's take a look at the data. The data are in data and in the community data folder. And I believe they are in iPlant training, which is favorited there, and in DE walkthrough. Okay, so I can open this file. Certainly in the discovery environment, I can preview many file types, plain text, tab limited, etc. But these are my unaligned files, which I'm going to use in a few moments to go ahead and make an alignment with. Excellent. Okay, so I'm going to close that and now go to Apps. Okay, so in Apps, I have a listing of all the bioinformatics applications that are currently deployed, or at least that I have permission to see. Right now it says 351. I can search for apps by name, or I can sort of look through and see if I am able to find them in a category. But I'm going to search for an app that I know is there called Muscle. Okay, and Muscle is an application that allows me to make a multiple sequence alignment. As you can see, I favorited this app, so actually on my listing here, it already is categorized under my favorites, so I could always find it quickly. If I'm not sure what an application does, I can click on the info button, and then I could see a little bit of the description. I could click and go to a user manual, so I have all that information available to me. In the tool information, I can see what's the version of the tool, so a whole bunch of things that might be useful. Additionally, if I like an app very much, I can give it a rating, gold stars. If I think it's not working, I can give it a lower rating. Most of these apps, in fact, these happen to be mine, so they're not rated by anybody. The apps, it's a community forum, so people can leave their opinions and comments, but we certainly would follow up on anything that we think is not working. In order to actually use the application, however, I just need to click on the name. So I'm going to go ahead and click on it. It's going to open a new window. I'm going to close this other stuff behind there. Okay, so all the applications within the discovery environment look very much like this. I have a name, and by default, I'm just going to go ahead and add demo to that. And I really encourage you, this is something that's really important. Remember I said reproducibility? So if you're like me, when you're using a new tool, especially a click and point through a graphical interface, you are pressing all sorts of buttons, sort of intuiting your way through how it's supposed to go. But then when it comes time to reconstruct what you actually did, how do you figure that out? So as we'll see, in many places there are opportunities, even though we're using a graphical interface, to be reproducible. So back to the screen, even though this is just a demo, I'll just say that here in the comments. This is a demo. So please be in the habit of actually leaving comments to yourself and documenting your work everywhere that you can. Okay, the next thing I'm not going to touch. It says select the output folder. I'm not going to touch that because I'm fine with the default. However, remember I said you could have a shared folder where maybe that's where you always output things. You could change that, but at this point, don't touch that. Now I need to select where is my data that I'm going to pull from. We already saw it, so I'm going to browse. And remember, it's in community data, iPlant training, DE walkthrough. By default, it opens to the last place I was. I'm going to go ahead and click on that DE sample plants and say OK. This is a very simple application because it only has one under the mandatory option, and that is the sequence type. And from my list, I know that that's DNA, so I'm fine with that. I can go ahead and click Launch Analysis. And that's it. Basically, any application in the discovery environment is going to look and function just about the way that I've shown you there. Now, to actually see what's going on, now I'm going to click this other button, Analyses, and I can see the status of what's going on. And you can see I'm getting updates on the bottom. The demo is submitted, the demo is running. On the upper right-hand side here, I have some status about what's going on. I can see folders that have been shared with me, data sets, etc. Okay, but what I wanted to focus on here was my analyses, and I can see that I have an application, Muscle is running, and so it tells me when it started, hasn't finished, so I've got nothing there. Let's actually look, see some of that metadata reproducibility stuff. If I click on an application I've run before, and I go up to Edit, or rather Analyses, I'm sorry, I can go and view the parameters. So I can actually see a whole bunch of information about this job that I launched. I can see, and I have links to, if I click any of these are live links. I could go to any of the files that I used as input. This is important. Maybe I didn't name my files with any sort of intelligible naming system. I can see all of the different values for different parameters. So I really do have that ability to be reproducible. I could save this as a text file. Let me go back up here. If I wanted to relaunch that job without filling in all of those parameters again, I can go up to Analyses and click Relaunch. And it's going to open that application, in this case Top Hat. It's going to have all my input data already there, my reference annotations, everything just as I selected it, but maybe I want to go through and change my anchor length to 6. And so rather than entering everything all over again, I can just make that one change and click Launch, and it will relaunch, okay? But I'm not going to do that this second. I just got a notification that it stopped. In fact, I need to refresh this. So my job is now completed. In fact, those are the status, running, complete. If there was an error, I would get that, or failed rather. But now that my job is complete, if I actually click on the job itself, or the job name, it's going to take me to the outputs where they exist in my iPlant data store. Okay, and there are my results. If I click on that output, here are my aligned sequences. Okay, so really, launching a job within the discovery environment is just as you've seen. You select the application, you can browse for those inputs wherever they exist within your iPlant data store, and there are hundreds of applications that are there. We of course have some tutorials to help get you started in terms of the science, but we'll get to that towards the end of the demo. At this point, I want to remind you, if you have any questions, please at the bottom, go ahead and chat me those questions. I'd be more than happy to answer those. Okay, so now we're going to move on from the discovery environment to actually cloud computing and what that is entailing. So let me go back to the slide to have a couple to orient us here. Okay. So Atmosphere, which is the iPlant cloud platform. What does that mean? How can you take advantage of that? So Atmosphere gives you one-click access to more than 200 virtual machine images, maybe more than that right now. It allows you to fully customize your software setup, and again, it's integrated with our computing and data resources. First, I want to answer the question, what is cloud computing, at least in our context, because there's a lot of advertisements and products, and what does it mean? Well, let me give you definitions for two important terms that I'll be using. The first term is image. What does it mean when I say I have an image? Well, we've all been, I think, through the situation where we have a computer and we buy a new one, and then we want to take everything that's on the old computer and put it onto the new computer. So from your original system, most likely you have some documents, some images, photos, movies, whatever, and you can put those onto a backup hard drive. But there is another type of file that you could create, and this file is called an image file. And an image file is unique because an image file is an exact copy of everything on that system, including the operating system. So whereas on the new computer, you'd have to reinstall Office, you'd have to reinstall particular programs to get them to work, with an image file, you can actually clone your computer. So on the new computer, documents are still documents, but image files really become a complete clone. Everything down to the desktop that the wallpaper that you have on your desktop and where the icons are. Okay, so that's what a complete clone is, or also known as, in this case, an image. Okay, so that's one piece of terminology. The second piece of terminology is instance. Okay, so in the iPlant cloud, we have hard drives, we have CPUs, we have memory, and when you come to Atmosphere to use the cloud, you're going to ask us for a certain amount of this. Maybe you know how much hard drive space you're going to use, how many CPUs you need, how much memory, and then you're also going to need an image, and your image is going to be, for example, a Linux system that has a certain software installed, maybe even has some data sets installed. So once you have the physical hardware plus the image, the software, and the operating system, that is your instance, or sometimes also called a virtual machine. So with Atmosphere, we can create an instance for you, custom to suit your needs. What is this useful for? Well, first let me talk about the specs. You can choose an existing image, or you can customize one that we have. You can launch up to a 16-core, 128 gigabyte machine. You can access it as we're going to via the shell or VNC, and you can even share your image with other users or even make them public. There are a lot of reasons to use this. You can run software and analyze data sets that you currently only run on your laptop, in which case maybe you have to give your laptop up for an afternoon while something is running. In the discovery environment, one of the deficiencies is you can't run a program that has its own graphical interface. But through Atmosphere, you can run programs with GUIs, everything from something like RStudio to some different bioinformatics packages that may have a graphical interface. You also have sudo access, which means you have administrative rights on your Atmosphere instance, so you can definitely install anything that you'd like. If you're in a setting where you need uniformity, a teaching setting, for example, you can run a whole bunch of instances to support a classroom. And also, if you're producing software workflows or data sets, you can make them available through an image to a larger user community. So I'm going to show you in a demo essentially how to connect to an Atmosphere instance. There are several ways. All the instances you can connect to via the command line, via SSH, using either PuTTY or your terminal. And many of the instances which have a desktop can actually be accessed, as we're going to do, through VNC. Okay, so let's do that demo really quickly. Come back to the iPlant Collaborative homepage. Okay, and this time what we need to do is where it says Atmosphere, you're going to go ahead and click on Launch. All right, now I already have some things running, which I'm going to show you in a moment, but what you'll do is you'll click Launch New Instance. And then you'll first see a collection of images that you can choose from to work with. So I'll give it a second for those to load. Okay, so there are a bunch of featured images. If you scroll through, you may see tags to the images that give you ideas of what might be available. I can see stacks, maker, image processing toolkits. I can see tags for phylogenetics, for BioPy, NumPy, genome annotation, etc. So you can really search through and see what is of interest to you. I'm going to go ahead, if I want to launch something, I could then rename my image according to what I want, and then I could choose the size. The caveat is, of course, not every single instance size is available at every single second. There is also some allocation, so you can see how much you'll be allowed to run based upon what you've run so far this month. And of course, if you need more resources, you can request more resources, but your allocation will be refreshed at each month. In fact, before you even come into Atmosphere, if anybody's trying now, you may see you don't have access. Let me remind you from the iPlant homepage that when you come to iPlant, you don't automatically have access to Atmosphere. So you're going to go to the user portal from the homepage where you created your account and log in. And then from there, you're going to see Atmosphere, except it won't be under My Services as mine is because I have access. It'll be towards the bottom under Available, and you can click Request Access and fill out the form. You must be registered with a .edu or .org email account. Okay, so back to Atmosphere. Once I select what I need in terms of those instances and my size, I would then click Launch Instance, and within a few minutes, I'm going to get an instance over on the left-hand side. When that instance reports that it's active and the color is green, I'm then able to connect. So I've already downloaded VNC Viewer, so I'm going to use that. But there's also, if I click on VNC, in fact, let's do it that way. If I click on here and say Access via VNC, it'll launch a Java application. So you could either download it, or if you have Java, that'll work fine. Okay, I don't need the update right now. Skip that, run that, and connect. I'm going to have to continue and then click enter my iPlant username and my iPlant password. Click OK. And now I'm connected to my virtual computer. This is a virtual desktop that is running, and it's just like another laptop or another server that I would have running somewhere. I could do any type of analysis on it that I'd be interested in doing. Okay. If I wanted to connect to this via the shell, via SSH, I can do that as well. Let me start a new terminal. Excuse me. And all I'll need is my IP address, which is back here. And make it a little bit bigger. And then I can do SSH. My iPlant username at my IP address and hit enter, and I can say yes to continue, enter my credentials, and now I'm connected. All right, if I want to, example, create a directory, and you should be able to see that on the left-hand side that directory is there. So I'm connected two different ways. If you do download VNC or you have that installed, you connect with the same IP address. You're just going to have to enter :1 after it. Connect. It's really the same thing on that standalone program. I don't know how valuable it is outside of the educational purpose, but you can see I can connect in a variety of ways to that remote computer. I could even share that computer with somebody else so they could see my desktop and work with it. Okay, I have a question from Brian about .gov registrants being allowed to access Atmosphere. I believe you should be able to, so just go ahead and email support@iplantcollaborative.org. So we do have a lot of users who are with the government, and they also have access to it, so you should be okay. Okay? All right, so coming back to Atmosphere, the last thing to know is that as you're running something on Atmosphere, it is going to spend down your allocation. Again, if you don't request more resources before your allocation is through, your instance will be shut off until you're refreshed. You can pause or stop an instance when you're not running it. And of course, once you're completely done with an instance, you're going to want to click Terminate. Now be careful, you want to transfer data off of that instance before, because once you terminate, you will not be able to recover anything. No one will be able to recover anything from that instance. So what could you do on that instance? You could go ahead and start CyberDuck on that instance. You could go ahead and use iCommands and transfer the data back into the regular iPlant data store. When your instance starts up, you actually don't have any of those data sets that are in your iPlant folders, so you'll need to transfer those in, analyze them, and then transfer anything back out once you're done. Okay? I'm done, so I'm going to go here and click Terminate. To terminate this instance, I don't need it anymore, and that'll kill the connections that I have going on elsewhere as that starts to connect. Okay? All right. So those are the demos that I wanted to show you. The last thing I want to show you is where do you go from here. We have the iPlant Learning Center, so I want to make sure you know where that is, because you have a whole bunch of resources there. So one last time back to the iPlant homepage. Down at the Learning Center under Get Started, if you don't remember anything else, go through the Get Started guide, and that would allow you to cover many of the basics that we did today. There's a whole bunch of platform and science tutorials. You may want to learn how do I use to assemble a genome with SOAP de novo, how can I do a GWAS, how can I use QIIME to do some metagenomics. There are a bunch of tutorials there. Very important from the Learning Center, and also you'll see links to it, is the ask.iplant. You can post questions to the user forum, and people will answer them. Also, on the homepage, you could see we have workshops. There's one that's coming up in a couple of weeks at UC Davis. So I encourage you, if you are in the area, please apply. I think there are some seats still available to the UC Davis workshop. You'll get a lot more training and hands-on. But with that said, we're at the hour, and I'll give a few more seconds for anybody to chat questions. If you don't have any questions, I ask you to stay on just a moment, because when the workshop or when the webinar ends, you'll be pushed to a survey where you can help us know if this was useful for you, or let us know about anything else you'd like to see from iPlant. So I'm going to give just a few more seconds down at the bottom here, see if we have any questions. Besides that, we'd like to say thank you to everybody who signs up for an account. We are definitely here to collaborate with you, to serve you, and we look forward to helping you accomplish your goals. And with that, since I don't see any other questions coming across, I'll encourage you to reach out to us on the ask.iplant collaborative forum, where many people, everyone in the entire community, is able to see your questions and put answers to them.